Hello everyone. Here we are discussing the one of the most important application of recombinant technology, that is the production of recombinant pharmaceutical, and we are taking the example of insulin here. Recombinant insulin production by the use of RDT or genetic engineering. So we all know the importance of insulin in our body. During fed condition, the concentration of insulin increases. Insulin is produced from the beta cells of the islets of Langerhans in the pancreas. So whenever one person is in fed condition, fed means the availability of glucose in the blood increases. So we need to transport glucose from the blood to the cell, and for this, cell may have several receptors known as the GLUT receptors. Maybe GLUT1 or maybe 2 or maybe 3 or maybe 5. These types of receptors are present on the surface of the cell. GLUT stands for glucose transporter, GLU means glucose and T for transporter. So these receptors are basically present on the surface of our cell so that we can transport glucose from the blood into the cell cytoplasm so that we can initiate the glycolysis process. And for this transport of glucose via this GLUT receptor, this receptor needs the participation of the insulin hormone. So if a person is in fed state, the level of the glucose, blood glucose increases, and in that case the islets of Langerhans, the beta cells of the islets of Langerhans of the pancreas will produce the insulin hormone, and the function of the insulin is to transfer glucose from blood to the cell cytoplasm. As a result, we can initiate the glycolysis process and ultimately we can generate some amount of energy. So if you think the structure of insulin is basically, mature insulin is basically composed of two chains known as the A chain and another is known as the B chain. But insulin is normally not released from in the form of mature insulin. Initially, it was released from the islets of Langerhans as a Prepro insulin form. If you can see the structure of the Prepro insulin, so this is the structure of the Prepro insulin, which contains A chain, B chain. A and B chains are joined by the C chain. C stands for connector chain, which joins the A and B chains. And additionally, a leader sequence is also available. Leader sequence is nothing but some amino acid which will provide the signal that this is the molecule that need to be transported. Okay. So the leader sequence is basically present in the structure of Prepro insulin. So from Prepro insulin, proinsulin will be formed. In that case, the leader sequence will be removed by the protease cleavage. So the leader sequence, which was initially present in the structure of Prepro insulin, now will, during the formation of the proinsulin, this leader sequence will be removed. As a result, this A, B, C chains are spontaneously assembled together to form a structure which is called the proinsulin structure. Here, if you consider this leader sequence is not there, if the leader sequence is, say, the leader sequence is absent, in that case you can say this is the structure of the proinsulin. The leader sequence is removed. So the function of the C chain is to make disulfide bond between the A and B chains. And C stands for connector. Connector means it will connect between the A and B chains. So once these A and B chains are joined together by the two disulfide interactions between A and B chain, in that case there is a no role of the C chain, and in that case we can remove the C chain. So cleavage occurs. As a result, now the C chain is getting removed. So the A and B chains are now joined together and it forms the mature insulin. So that means when the islets of Langerhans first releases the insulin molecule or first produces the insulin molecule, it is in the Prepro insulin form where the leader sequence and the C chains are attached. And then whenever it forms the proinsulin, in that case the leader sequence is getting removed. The leader sequence only the only amino acid that is essential for the transport process to occur, that it should be released into the cell cytoplasm and from outside. And the thing is, the after the formation of the proinsulin, which contains the ABC chain, or you can say this ACB chain, okay, the C chain is also present. The C chain, the function of the C chain is to join between the A and B chain, and when they form the disulfide bond between the A and B, due to the presence of cysteine residues in these two polypeptide chain, and then there is a nodal of the C chain, so we can remove the C chain. Protease cleavage actually cause the removal of the C chain. As a result, mature insulin will be formed. So whenever you need to biosynthesize this mature insulin in our body, not in our body, sorry, in the bacterial cell, that is we want to make some recombinant insulin. In that case, we have to consider the A chain and B chain only. The focus about the A chain and focus about the B chain. What is the size of the A chain? A chain is composed of 21 amino acids. So as you can see, A chain is composed of 21 amino acids. So we know the sequence of the amino acid present in the A chain. And the size of the B chain is about 30 amino acids. So we know the sequence of the amino acid present in the B chain. So we can synthetically made the nucleotide from these amino acid sequences. For example, if we know the A chain starts with the methionine residue, in that case the corresponding codon will be the AUG codon, and in that case you can easily make the sequence of the DNA present. So this is the amino acid, this is the mRNA codon, and corresponding the sequence of the DNA can be easily manufactured. Okay. So what we need to understand, that if we know the sequence of amino acids successively in both A and chain and B chain, in that case we can synthetically manufacture the DNA molecule corresponding to the A chain and B chain. So now the strategy is, in order to obtain the recombinant insulin, the strategy is to synthesize the artificial genes for the A chain and B chain, followed by the production of the fusion protein in E.coli cell. So this thing is illustrated in this particular diagram, that now we have a synthetic A chain and now we have a synthetic B chain, and these individual A chain and the individual B chains are fused with a vector. We can take vector, any kind of vector, because the size of the chain is very small. So you can take any plasmid vector like PBR322 or maybe PAC18. You can take any of this vector, and now the A chain, before the start of the A chain, the LAGZ gene is attached. Some modification has been done. That is, the A chain, you have the A chain, say this is your A chain, and whether this A chain is present or not within the cell during the cloning experiment, you need to add some kind of selection vector, and we have added the LAGZ gene as a selection vector here, LAGZ gene. So this is the LAGZ gene, which can produce beta-galactosidase enzyme, and between these two, that is the A chain or maybe B chain and the LAGZ gene, we have added a methionine. Okay, so there must be some sort of methionine present between them. So this is little modification that we need to make. Why LAGZ gene and what is the importance of the methionine? We will discuss this thing definitely. So say this is a promoter, which is the LAG promoter. So if it is a LAG promoter present, and if we can ligate this whole assembly into that particular portion, in that case the LAGZ gene will be expressed definitely, and the cell will be able to produce beta-galactosidase enzyme. And any cell that is able to produce the beta-galactosidase enzyme, they cause the expression of the particular gene of interest, right? So if it is the A gene, so this cell, the cell that have that particular recombinant DNA will express the A gene together with the beta-galactosidase enzyme, and the cell which carries this particular B gene, they will simultaneously express the beta-galactosidase plus the B gene product. So we have taken this thing into E.coli cells, say artificial transformation is done. This recombinant DNA, after making this recombinant DNA, artificial transformation can be conducted so that this recombinant DNA can be transported into the E.coli cell, and now the recombinant E.coli cell is inoculated in a suitable broth, so that if the recombinant cell is present, they will release the A gene product. the product of the A gene, that is the A chain actually. A chain together with the beta galactosidase enzyme plus the methionine portion is also available. And if you think about the B conical flux, in that case the B conical flux will produce the B gene product together with the beta galactosidase and the methionine portion. So during the cloning experiment we need this beta galactosidase enzyme so that we can check in which cell the product is getting expressed or not. But after product formation we don't need the laxate gene and we don't need the methionine portion. Here the importance of the methionine portion arises. So if you have the protein, you can see this is a protein that we can isolate from this conical flux. The protein contains the A chain plus the beta galactosidase segment, and now they have there are a methionine segment between this beta galactosidase enzyme and the A chain, and simultaneous phenomena can be also observed in the segment number B, that is the B chain composed of 30 amino acid. There is a methionine residue before the start of the B chain, and before the start of the methionine residue there will be one beta galactosidase gene or enzyme. So we need to remove this segment, and now we have treated this sample with the cyanogen bromide CNBr. Cyanogen bromide can selectively cut the carbon terminus residue of the methionine molecule. Once again, CNBr actually cleaves the C terminal region of the methionine residue. As a result, they will cut this methionine segment. As a result, this beta galactosidase together with the methionine will be eluted out. Only the A chain will be available, and simultaneously this beta galactosidase plus the methionine segment will be eluted out. Only the B chain will be present. So we need to purify this A chain and B chain, and then we can artificially make the disulfide bridge between the A chain and B chain. In that case, what we want to say, that is the E. coli cell, from E. coli cell or from any bacteria, we cannot produce the total recombinant protein or active recombinant protein according to this diagram. According to this explanation, one E. coli cell is producing the A chain, another E. coli cell is producing the B chain, and then after isolation of this purified A chain and B chain, we need to make the disulfide linkage between the two A chain and B chain so that the mature insulin can be biosynthesized. So this is the major application, that why we need to add the methionine residue between these two chains, and what is the importance of the beta galactosidase segment here, so that we can check that in which cell the specific gene of interest is present and in which cell the specific gene of interest is not present. Because whenever you have added the beta galactosidase segment, we can simply do the XGAL plus IPTG, so that we can select the blue head screening, that which colonies are blue and which colonies are white. If the colonies are blue, in that case you can say the beta galactosidase segment is available. If the colonies are white, in that case you can easily select that the beta galactosidase segment is not available. So the segment of beta galactosidase is absolutely important, and also you need to add the lac promoter here so that the beta galactosidase segment can be transcribed, and also your gene of interest can also be transcribed. So this is all about the basic procedure by which we can artificially manufacture the insulin hormone in bacterial cell, although not in active form, because one E. coli cell is producing the A peptide, another E. coli cell is producing the B peptide, and we need to isolate them and we need to purify them, and after that we need to ligate artificially in vitro system. So E. coli cell is not actually actively producing that particular thing. There is another way by which you can manufacture this thing. One can easily say that you are actually transferring the A gene in a particular E. coli cell and transferring the B gene in another particular E. coli cell. So in two different E. coli cell, two different genes are present, but the size of the genes are very much small, because A gene is composed of 21 amino acid, that means it should be 63 nucleotides actually, and B gene is composed of 30 amino acid. B gene is composed of 30 amino acids, so 30 amino acid means approximately it is the 90 nucleotides. So 63 nucleotides plus 93 nucleotides, not a big problem, because if we consider any plasmid DNA, they can actually take like PBR 322, they can take up to 4 kilobase pair of DNA or maybe up to 6 kilobase pair of DNA. So if we consider 4 kilobase pair means 4000 base pair, so it is a very large segment compared to this section. So one can actually think that okay, I can construct some vector. I can construct some vector which contains the say it is a vector like PBR322 which contains the lac promoter here. Okay, this is the lac promoter, and then the beta galactosidase segment should be present. Okay, this is the beta galactosidase segment, beta galactosidase gene, so that we can determine that in which cell the beta galactosidase is expressed or not. And followed by what you can do, you can add the A chain, A gene, then the C gene also. We need to add the C gene and also the B gene, because the segment of each of these genes are not very large, followed by the remaining portion of the vector. In that case, the one E. coli cell is actually producing there must be some sort of methionine between the beta galactosidase and the start of the A chain definitely. So that say this is the beta galactosidase segment followed by the methionine residue, then the three product will be there, that is the A gene, then the C gene, and then say this is the B gene portion. Okay, so this is the A gene portion, this is the C gene portion, and this is the B gene portion. In that case, if you can easily select that in which cell this particular DNA is incorporated, right? Because if the cell have the beta galactosidase gene, they also have this A, C, B gene clusters. And if the cells have the beta galactosidase gene, they will express the beta galactosidase enzyme. So we can add XGAL into the media plus an inducer like IPTG, isopropyl beta thiagalactoside. In that case, this beta galactosidase, if present, they will metabolize the XGAL to produce a blue colored compound. So if the blue colored colony appears in your Petri plate, in that case you can easily say, if the blue colored colony appears, you can easily say that that is the particular cell where your gene clusters is there. And if white color colony appears, in that case they do not have the beta galactosidase gene. So after purification of this segment, what you can do, you can easily cut this methionine portion together with the beta galactosidase segment by using cyanogen bromide CNBR treatment, cyanogen bromide treatment. So now you have ACB3 chains, and they will spontaneously assemble. And as a result, say this is the A chain. Now the connector chain is also available, C chain, and now the B chain is here, somehow here. So they can make some disulfide bond, two disulfide bond between them. Okay, so say this is the disulfide bonds that may appear, and after that you can purify this segment, that is the pro-insulin from the E. coli cell, and last the peptide protease cleavage to remove the C chain to make the mature insulin. Okay, you have to make the mature insulin. Mature insulin is composed of only the A chain and B chain, but the bacteria is producing the ACB chain. That means it is in the form of pro-insulin form, right? It is a pro-insulin form or inactive form. So we need to take out this particular thing from the bacteria, purify this thing, and ultimately remove the C chain so that the mature insulin will be formed, and that is the active insulin. Okay. So once again, from this experiment we can actually conclude that if anyone asks you that whether the bacterial cell produces the mature insulin or not, or active insulin or not, the answer is no. The bacteria is not able to produce the active insulin. Rather, the bacteria is able to produce either the pro-insulin form or the inactive form totally. Because if we think about the first diagram, in that case one bacteria is producing the A chain and the bacteria is producing the B chain, and you need to purify this thing and you need to ligate this thing. So in two cases, both of these cases, we cannot actually biosynthesize the active insulin in the bacterial cell, but more or less major parts have been done in the bacteria, and then you need to purify this thing and ultimately make the mature insulin in vitro system, right? So this is all about how we can make the insulin or recombinant insulin bacteria. In the same way, we can also make the human growth hormone, like if you think about the somatostatin hormone, it is a very small hormone, human growth hormone. Somatostatin is a very small hormone, human growth hormone, composed of only 14 amino acid in length. So if it is only 14 amino acid length, it is very simple, and there is no such chains like A chain, B chain and C chain, only one chain is available. So 14 amino acid means, that means 14 into 3, that is equal to 42 nucleotide actually, and you can tag one methionine residue before the start of the gene, and also the beta galactosidase enzyme, beta glucosidase gene, and do the same process by which you can manufacture the insulin protein, at least the A chain. In the same way, you can manufacture the recombinant somatostatin hormone. Why you are saying recombinant here? Because we need to manufacture the nucleotide sequence. We need to make the nucleotide sequence on the protein sequence. And we all know that, say, if your protein contains serine amino acid, serine means, say, UCU. Serine also means UCG. Serine also means UCA. Serine also means UCC. So the third codon, which is a Ubbel codon, that can change from U to G to A to C, anyone. So that means you don't need to go for the UCU. Say UCU is actually present in the protein sequence, but you don't need to manufacture the UCU. You need to go for the UCG also, you need to go for the UCA also, you need to go for the UCC also. You can take any codon about the serine and you can manufacture the gene sequence, okay, nucleotide sequence, because whatever the nucleotide sequence that you have made from either UCU or UCG or UCA or UCC, they will ultimately produce some mRNA, and that can code the serine amino acid. So it is not essential that in physiological system the sequence of that particular DNA present for the production of insulin or the production of somatostatin will be absolutely same that we have made in artificially in bacteria or by this technique. Okay, so somatostatin hormone can also be produced by this same procedure. The strategy used was the same as described for the recombinant insulin, involving insertion of the artificial gene into the lag Z prime vector, synthesis of a fusion protein, and cleavage with the Sarnajan membrane. So this is the absolutely same procedure. Okay, so this is all about how to manufacture human growth hormone like somatostatin hormone, which is a 14 amino acid in length, and insulin hormone in bacteria by the help of recombinant DNA technology.